So welcome everybody. This is a virtual workshop on new DNA repair capacity methods developed for clinical and population-based studies. This was originally intended to be a pre-conference workshop at this August ISEE meeting, but since that went virtual, we are instead doing all workshops during the year. I would like to introduce one of the organizers of this workshop, Dr. Dan Shah. I'd never asked you how to pronounce your name. Sorry, I just realized that. Shaughnessy?
It's Shaughnessy, yeah.
Okay, perfect. Thank you. Who is a program director at the National Institute of Environmental Health Sciences in the Exposure Response and Technology branch. So with that, Dan, thank you.
Thanks, Marianthi, and welcome to everybody to this virtual workshop. I'd like to thank the organizers, Marianthi, and then also Joan Casey and Molly Kyle in particular for kind of seeing this through. These are interesting times. You know, we've had to be very creative in holding any kind of meeting or workshop. So this is kind of a trial run, I guess, in how to do things in the current times. So you've all had a chance, hopefully, to view the presentations from the 5 speakers that will be on today, as well as the overview and introduction that Gwen Coleman here at NIEHS gave. So as Gwen described, there have been significant advances over the last, I'd say 30 years in our fundamental understanding about how cells respond to outside or stress and damage to DNA, both from outside stressors and then as a function of kind of normal metabolism. So without efficient repair and replication systems, we'd all be in big trouble. And you can see that in a number of diseases and syndromes where people are deficient in repair enzymes or function. So translating this fundamental knowledge into practical ways to measure an individual's ability to respond to and repair damage for population studies and clinical studies has really been hampered by the lack of high throughput, cost-effective, easy to use assays. And so we're going to hear today about some exciting new approaches from the 5 speakers here for measuring DNA repair capacity that ultimately can be scaled up to large numbers of people. Again, we'll hear from the 5 speakers. To kind of remind you of each of their presentations, I've asked them to give maybe a 2-minute summary of what they're describing and what they're working on in these new methods. And then we'll have a chance to get all of your questions and comments, which is and feedback for these, which is really the impetus for this. Early in the development of these kind of new technologies, we really like to hear feedback as things are being developed, how you would use these, what you see are the other challenges that maybe the developers haven't really thought of yet. So in order to do this, you can either type your question in the chat box, and then if you look in the participant box, at the bottom there's an invite at the left, a mute me, and then there's a 3 little dots icon. If you click on that, you'll see a raise hand icon, and I'll call on you. So we'll just do specific questions for each presenter, and then maybe if we have time at the end, we can kind of cover more general topics for this area. So that's all I had to say. I don't know if there are any questions about the logistics. You know, just put something in the chat box if you're having any trouble. But with that, I'd like to start with the first presenter, who is Zach Nagel from the Harvard School of Public Health. So Zach, the floor is yours.
Okay, thanks Dan, and thanks Marianthi and everyone who contributed to putting this together, and all of you for showing up. So again, I'm Zach Nagel, and like all of my colleagues here, I'm interested in the molecular basis for susceptibility, excuse me, to diseases that are caused by compromised genome integrity and often caused by DNA damaging agents. So I think you see that there are quite a few trends that run through all of our science that are in common. And so I won't belabor those because I think everyone has a similar viewpoint on a lot of these topics. However, what was kind of the focus of my presentation was an assay that we developed that's called fluorescence multiplex host cell reactivation. And this is a plasmid-based assay where we introduce DNA damage site-specifically into closed circular plasmid DNA. And these plasmids have fluorescent reporters such that after they're put inside of cells, if the cells are able to repair the damage, we get a fluorescence signal that tells us that the damage was repaired. And the advantage of this technique is that it's fast. We have control over the type of DNA damage that we're measuring, that we're repairing. And it's multiplexed, so we can use different fluorescent colors to measure each of the different DNA repair pathways. And so the basis for the assay, as I mentioned, is generating these reporter plasmids in vitro, introducing them into cells, and then running a flow cytometric assay to measure fluorescence signal, which tells us about the DNA repair. and the two areas where we're really applying these are in population studies primarily. So we're interested in differences in DNA repair capacity that may predict disease risk or sensitivity to exposure to DNA damaging agents. And another area that I touched on later in the presentation had to do with measuring DNA repair in tumors, since a lot of tumors are treated with agents that damage DNA. That's the mechanism of action. So understanding tumor DNA repair may help us to predict whether a drug is going to be effective. And so I think that's more or less the summary of my presentation in a nutshell, that we have these fluorescent assays, and we're looking to apply them towards personalized medicine and personalized prevention in the context of populations. So I will look forward to answering any questions, and thanks again, everybody, for being here.
Thanks, Zach. So any questions, you could just unmute yourself. Maybe while people are thinking about it. Zach, and you spent a lot of time in the first couple of years of this trying to figure out the individual white blood cell types that are most informative for looking at repair. Are you thinking ultimately you'll have to kind of settle on like B lymphocytes or T lymphocytes, or will there be a possibility of kind of doing a first pass with whole blood, you know, like Buffy code or something like that, and then drilling down?
Okay, yeah, thanks, Dan, for that question. So I think it's important to know which cell type we're looking at. We're not sure yet whether every cell type will tell us the same thing about inter-individual variation. I think this is a big open question in the field. Since blood is usually what's available, that's how often these studies are often done. And the underlying assumption to that work is that whatever you measure in the blood cell is going to be predictive of what also happens in other tissue types. One of the first ways we're trying to test that is just to see whether person-to-person differences in T cells, which are most commonly used and easiest to work with, are representative of inter-individual differences in other types of cells. And we don't have a verdict on that, but our assays, being cell-based assays, we can use surface markers to label the different cell types in PBMCs. And that allows us to measure repair as a function of cell type at the same time as we're doing our fluorescence-based assays. So I think it's possible that it'll be most informative to have information about more than one cell type. It would probably be ideal if we had tissue cells from the tissue that we're really focused on. So if we're studying lung cancer, the best thing would presumably be to look at lung tissue. But if we can at least show that there are correlations between the DNA repaired person-to-person differences across tissues, then it would at least validate blood as a useful surrogate for what happens in other tissues.
Thanks. And then we have a question in the chat box from Kristen. I guess that's related to my question. Can you use stored or fresh blood samples? Does the assay work on both, or just prospectively collected samples? In other words, only prospective PBMC collection or something else?
Okay, yeah, thanks for that question. So that touches on a number of the things that I and others in this group are working on as part of the consortium that we're involved in. The FMHCR assays, we've seen that they work certainly on fresh blood cells, whether they're proliferating or not. And they also work on previously frozen samples. And one of the questions we're addressing is how well the previously frozen samples reflect what the fresh samples would have looked like. In general, for epidemiology, it's not feasible to work with fresh samples, although you could consider having someone overnight the blood to you. And that may be one way to do it. But the easier thing, at least from a laboratory perspective, is if we can freeze down the samples and recover them in large batches. And then if there are batch effects, at least the batches can be large. If batch effects are significant and it's person to person, then we're getting a batch. One person is one batch, so that wouldn't be ideal. So what we're doing now is a repeated measures study where we're going to have 12 people who donate blood about once a month for a year. And we're going to measure DNA repair in the fresh cells, freshly isolated cells, as well as those cells after they've been through a cycle of freezing and recovery. And so I think that will answer a lot of these questions, which are extremely important.
Kristen, did you have any follow-up, or did that answer your question?
No, that's it. I just didn't know how far the assay development was in terms of the feasibility of actually integrating these metrics into cohort studies right now, or prospective studies right now.
Yeah, maybe the one thing I'll add too is the data I presented in the talk were all from previously frozen lymphocytes, so that had been recovered from cryopreservation. Mm-hmm. Our preliminary unpublished results suggest that there's actually more reproducibility if the cells didn't get frozen down. So it would only get better if we could look at fresh cells, but it does seem that we're able to make reproducible measurements in previously frozen samples. The only thing is they have to be stored in a pretty special way, so slowly cooled in the presence of DMSO with FBS. And most often what people do is snap freeze for DNA isolation. So that's a big barrier on a lot of studies, but here and there you find cohorts where samples have been preserved. I think maybe I should ask for clarification. One of the questions was about prospective collection, and I think this touches on another issue, is reverse causality. So if you do a study where the people are sick now and you want to understand if they, their DNA repair explains the risk of disease, it would be much better if we could get blood from way earlier. Mm-hmm. And so that, of course, the challenge there is if we start that study today, we won't know for 10 years, and we'll have to sample, a very large number of people to have enough who will actually develop the disease that we're interested in. There are a couple of studies out there where people have been prospectively banking samples so that you could kind of do a nested study where you go back and find the people who ended up getting sick and comparing them to people who didn't. I think that's the ideal scenario, as long as long storage doesn't alter the properties of the sample. So that's one of the other things that I think I and others are grappling with is how much we can rely on a long-term preservation of samples to hold up.
Right. Yeah, I think that my question went to how useful are all of these bank samples that are out there for use of these newer DNA damage biomarkers versus how much of this do we have to design new studies to move forward in terms of really being able to use this as a biomarker?
All right, thanks, Zach. I was going to ask a more general question, but I think I'm going to leave it towards the end. And it has to do with ideas that you've had about combining your FMHCR functional assay with a lot of other measures of damage and mutation, and why does this battery have to be to really get a snapshot of somebody's kind of repair status? But anyway, we can maybe cover that later on. So maybe we'll move on next to Mary Beth Terry, who's at Mailman School of Public Health at Columbia. Just a quick overview.
Sure, of course. And that was a great transition, Kristen, your question and Zach's answer to our project, actually. So thank you for that question. So my talk was combined with my colleague's talk, Dr. Wu, who will speak next. So together, the two of us with our other colleague at Columbia, Dr. David Brenner, our grant has specifically focused on looking at Dr. Brenner's high throughput assay, both for newly collected samples, but also the heart of the study is really to get at this question of how well does phenotype predict cancer risk years later. So we are, for that, really addressing a question of how well do these archival samples in our breast cancer family registry predict cancers as we move forward. For this grant, it's breast cancer, but we have other common cancers recorded within our cohort, and we followed individuals on average about 10 years. So one of the thrilling parts about being part of this collaboration is really to think about moving forward for cancer risk prediction, the value of these phenotypic assays. As you know, there's hundreds of genetic variants involved in DNA repair, important to all cancers. And the reality is we can create, polygenic risk scores that get at those genetic variants and combine them. Breast cancer, for example, has a polygenic risk score that, is involving about over 300 genetic variants. However, the phenotypic assays, at least in some of the older studies that have been done, have a much, much stronger magnitude of association on the scale of 8-fold, 10-fold, 15-fold. So these kinds of ratio measures in epidemiology, are unmet with anything else except for smoking, right? So the advantage of having a phenotypic assay for risk prediction is huge. So as the work from all the collaborators in this consortia, as the evidence is built, the real potential is for having some of these high throughput assays be something that could be rolled out as a way of identifying higher risk individuals earlier to initiate cancer screening earlier. And as you know, although we have effective cancer screens in common cancers like breast and colon, we don't start those until now for colon for age 45. That's been moved. But just from the data that the cancers across the board, but particularly colorectal cancer, has been increasing really dramatically in young adults under 40. So we need a way to identify them. It's not going to be through family history because the trends are moving too fast for family history to be causing those trends. It has to be environmental, but we have to have a way at the clinic to screen people to understand who would be more susceptible. So again, a polygenic risk score may be one way, but the GWAS studies that have been done, polygenic risk score, there's some correlation between these genetic variants and phenotypic assays, but it's not huge. So a phenotypic assay that has a much larger association with cancers would really be a game changer, in my view, to get people who can be identified earlier for, for screens that we don't have population-based screens until much older ages. So for our specific project, we are focusing on the double-strand break repair pathway, but other pathways can be investigated. But the bulk of the work we're doing now is really through Dr. Brenner's lab in showing that these archival samples that we have can be revived and used to do these prospective validations. And I'll let Dr. Wu tell you some of the laboratory work that she's been heading up.
Yes,
do you want to go ahead, or are there any questions for Mary Beth in particular? I actually had one, Mary Beth, and maybe you've covered this before, but have you used the double-strand break assay that David's developed to look at specific risk alleles in patient blood samples for, let's say, for breast cancer?
Yeah, so we're doing that now. Our new data collection was shut down a bit for COVID, but we are, David's idea, which I think is brilliant, hoping we can test it formally, is to do this in real time in patients to really kind of titrate's the wrong word, but make a decision about the amount of radiation to give someone to reduce their contralateral breast cancer risk. So that is something that is kind of a big vision, but slowly is being kind of tested.
Any other questions specifically for Mary Beth? Otherwise, maybe we'll wait until Wei Chen has kind of presented hers, and we'll take them all, the rest of them. Okay.
Hi. So my talk is mainly focused on give everybody a little more detail on how our assay has been developed. So in our research group, we have developed double-strand break repair phenotype assay using the immunochemistry approach, and also right now with Dr. David Brenner on the gamma H2AS flow cytometry approach to measure the individual's DNA double-strand break repair phenotype assay. And we also have a nucleotide excision repair phenotype assay that have been previously shown associated with breast cancer risk. And right now we are working on trying to modify the assay using much more safe and UV exposure, and then quantify at different time point to get a better accurate estimate on the individual's DNA repair phenotype. And I also, in our lab, we also focus on looking at the DNA methylation, because what Dr. Ningo has been mentioned about, because a lot of the DNA repair phenotype assay have been used in the crop preserves PBMC, and it's actually a mix of the different cell type. So it's different people have different distribution of the blood cell type. So how that will affect our measurement on individuals' DNA repair capacity is still unknown. So my labs have been trying to figure out if the DNA methylation will be related to the DNA repair phenotype, and also eventually affect the different cell type DNA repair phenotype and function. So that's all.
Great, thanks. Any questions for either Mary Beth or Wei Chen on their work? I know it's hard because maybe it's a while since people have seen the videos and the details aren't there, but I was wondering, I know that you're doing global methylation studies as a part of this project. And I think I've asked you before, have you picked up any specific methylation of DNA repair genes or response genes, for example, mismatch repair genes or things like that that might be affecting function?
So our we are using the next generation sequence. So our collaborators and Dr. Bentayko and his lab can use right now we have been set up to profile about 180 amplicon in one single library preparation. So we mainly focus on cover the double-strand break repair and also some of the other different DNA repair pathway. And because the advantage of using the next generation sequence is we can also adjust the genetic variants. So when we looking at the DNA methylation profile, and we are focused on the genetic variation that in that region, so we can able to see the DNA methylation effect and also including the genetic effect on the DNA repair pathway.
All right. Any other questions? You can just unmute yourself if you have any questions or comments. Well, if you want to keep thinking about any questions, why don't we move on? The next presentation that you viewed was from Laura Niedernhofer, who's at the University of Minnesota. So Laura, floor is yours.
Thank you very much, Dan. And for everybody who's participating today, we're really grateful to have this interaction. I'm a biochemist by training, and so I need all the help I can get from epidemiologist colleagues. So I would welcome the opportunity to work with you. So we're focusing on measuring just one DNA repair mechanism, and that's nucleotide excision repair. And the reason is twofold. One is it's quite easy to do because all you have to measure is unscheduled DNA synthesis elicited by UV irradiation, and that is pathognomonic for this singular pathway. So it's very easy to focus on that. And the other reason is complete absence of this pathway comes with a 10,000-fold increased risk of skin cancer, as well as a 25% chance of having neurodegenerative diseases. So we feel there's just a lot of room there to find subtle defects in nucleotide excision repair that could be affecting broader populations other than just folks with a diagnosis of a rare genome instability disorder. So we've made the assay very simple. We can do it on fresh blood cells, and we just use PBMCs, but we can apply it to any viable cells that you would provide to us, and in a very high throughput flow cytometry assay. And what we're excited to do is to be able to, for the first time, ask questions about whether nucleotide excision repair capacity is heritable, if it's affected by environmental genotoxin exposure, if it predicts skin cancer risk, risk of neurodegeneration, risk of accelerated aging of any sort, or if you're a super repairer, maybe you're going to be a centenarian. So I look forward to working with any of you guys that can help answer those questions with populations that you're working with. Thank you.
Great, thanks. Any questions for Laura on the assay, the UDS assay that they've been developing? So Laura, when you presented this, you're looking mainly in lymphocytes with the new modified unscheduled DNA synthesis assay, but you're also comparing it to fibroblasts as well?
Absolutely. So what we're hoping to do is to establish whether or not peripheral blood cells are a good surrogate for keratinocytes as well as fibroblasts, because keratinocytes would be really the cell of interest in terms of skin cancer. If we have the opportunity, we would look at primary neurons and astrocytes and other things that could lend themselves to neurodegeneration, but that will probably be with mouse models. So yeah, eager to apply this to a variety of different cell types. But the primary goal was to make it easy by just using fresh blood.
And then have you looked at different types of skin cells, kind of from epithelium to fibroblasts to even stem cells, to kind of verify that you're seeing the same kind of repair kinetics in all cell types?
We have not completed that yet. We've got some preliminary data that suggests that repair capacity is quite similar and uniform in an organism. But I think this consortium opportunity gives us a better chance to really thoroughly test that in a proper way with human samples.
And last question that I had was, I think maybe Mary Beth, you touched on this in your, kind of the cancer statistics, but there's a rising population of melanomas, I think in young women, like in their 30s, which you might suspect is like use of tanning salons or things like that. But is that a target population where you could look at the genetics of maybe NER in those individuals to see if there's a genetic component, or is it mostly exposure, or it's a combination of both?
Well,
I couldn't agree with you. Oh, sorry. I couldn't agree with you more, Dan, and Mary Beth can follow up on that. But our first goal was to test patients that are coming through high risk skin cancer clinics. And if we have a patient survey that they fill out that indicates if they really don't think they've had a ton of exposure, if there is a family history also of skin cancer, and just trying to drill down in a superficial way that I think Mary Beth could address much better with genetic studies, if there is in fact a predisposition that's genetically linked. Sorry, Mary Beth.
No, I apologize, Laura. And anytime you need an epidemiologist, I'm happy to join. Your study is so important clinically. I think it's phenomenal. But yeah, just very quickly, it's like anything else, Dan. It's both. It's like the genetic susceptibility has to be there, but what's driving the trend is the environmental changes. So yeah, so it's definitely both, and that's where I think all of these studies, as they get high throughput methods, could be something very useful, because it's germline genetics, right? But then it's also phenotype and some epigenetic changes.
Any other questions, comments for Laura? If not, we'll move on. The last presentation that we had a video on was from Rob Sobel, who's at the Mutual Cancer Center in the University of South Alabama.
Well, thanks, Dan, and good morning, everyone, or maybe afternoon for some of you, and I appreciate the opportunity to be part of this workshop. It's been kind of fun. And so the talk that I gave, which is in line with the project that we're working on, utilizes a very classical assay referred to as the COMET assay to measure directly any insult to the genome. And again, like most of the other studies, we're focused on a donor or patient isolated peripheral blood cells, and both fresh or stored samples. But over the last 10 years, we and others have helped develop a more user-friendly approach to this COMET assay, and it's referred to now as the COMET chip system. So instead of running one sample at a time, we can actually run and analyze in a single day a 96-well plate of samples. So that gives one an opportunity to really start to think about this from an epidemiological viewpoint, since we can take either 96 different wells and evaluate it under 96 different situations, or we can probe many different patient samples at a given time. Particularly for our project, we're looking at donor-derived PBMCs. We've, like some of the other discussions this morning, we've looked at freshly isolated and as well as stored samples. So far, at least, we're not seeing big differences, which is good, because it certainly would be easier if these samples can be isolated and stored and then sent around the country for analysis. And then our goal, though, is really to ask questions of whether or not these cells, when challenged, if you will, respond differently. So in other words, are there functional differences among the 5 or 6 classically named DNA repair pathways? And to be able to understand that, we're in parallel evaluating a library of so-called DNA knockout T cells, as well as other libraries that we have to ask and answer the question, how does a DNA repair defect in this pathway, in pathway 1 or pathway 2 or pathway 3, or even gene 1, 2 up to 50, how does that give a different response in that assay, so that we can actually uncover, is there a functional defect in a particular pathway or a particular gene family within those donor-derived or ultimately patient-derived samples. And then finally, our focus is to even enhance the system further and to develop it as a 384-well plate system, which is an ongoing effort.
Thanks, Rob. So I guess I'll start again with the questions. So you're using a community-based study for your development. Are you, I think I've asked you this before, but are you genotyping the donors?
Well, that wasn't part of the funding resources, but all those samples are stored, and so we can do that.
So once you see maybe an outlier or something like that, you might be able to go back and look at a genetic reason for that?
Right. So we've isolated the DNA of every donor. And then, of course, since we're storing the cells, we can go back and isolate RNA, although in a lot of cases, we've already got that all prepared. So if we see any alterations in response, so far, they've all looked pretty within the norms. But that would be the goal, then to go back and reevaluate. And since we have, and we've now sort of done, like a lot of others have done, optimized storing these samples so they can be reanalyzed in a second and third time, and even amplifying the stocks from the classic 1 or 2 million to, in some cases, 50 million stocks. So we do have a good resource as we're progressing now.
And are they collecting family history of disease in the donor
They
system?
are. So everybody that participates, they go through a full medical workup and a full questionnaire, and we're trying to put all that together now in the first from the first groupings. Yeah.
So, any questions for Rob? A number of the investigators that have spoken are using the COMET chip technology, which, and I think Zach Nagel is using it as well, as kind of a measure both of basal level of damage, but also kinetics of repair, and sometimes it's used in combination. So Zach, what I was asking you at the beginning, you've kind of speculated that within the, kind of bounds of feasibility and cost, we probably are going to need multiple measures for an individual. So yours is a functional measure of repair capacity. In a sense, COMET is too, but it's also a measure of repair, and then mutational analysis, which we really haven't covered. So what are the consequences if you have low repair? You would assume that that person would have a higher mutation burden. So you have any thoughts kind of on where you see this going in the future in terms of this snapshot of an individual's repair and damage?
Yeah, thanks. I think this is going to become a sort of a computational challenge at the core. So certainly one of the things I didn't mention about the HCR assays is the limitations. So this is being a plasmid-based assay. It's on a small piece of circular DNA, which is not very much like the chromatin that you would find in the nucleus. And the COMET assays that Rob was just talking about measure repair on the genome, on the actual DNA that's already in the cell. So I think these are important complementary approaches. And then beyond that, many of us are applying more than one technique. So we have transcriptional profiling and SNP arrays. And ultimately, I think what I think we might have to do is something like in the way that Mary Beth Terry talked about a polygenic risk score for predicting disease. There might be scores that you would need that would take all of these inputs, integrate them into one readout that tells you something about the outcome that you want to understand. But also, I think, and this may be more of a mechanistic question than an epidemiology question, but I'm very curious how all these things will fit together. So it makes sense if you have lower repair capacity, you can have higher mutation rates, but I don't think we have a clear sense for how that should work out quantitatively. Is twice as much repair going to give you half as many mutations, or will it be a nonlinear relationship? And so I think all of these metrics at the end have to be integrated, and it's going to be, I think, a learning curve for us to figure out what's the best way to do that.
Yeah, I mean, as a follow-up, Dan, I would also say, as you notice, some of us are just doing this sort of globally. Some are looking, like Laura's, very specifically. So I think these are not going to be one-stop shop efforts, right? It's going to be very defined and maybe uniquely tailored to what the questions are being asked in whatever study is ongoing.
And Laura, I think you presented some preliminary data showing that there is, at least in the folks you've looked at, a huge difference. So that there are some like really great repairers, and at one end, and I know it's a small sample and early on, but I think across a lot of these assays we're seeing quite a lot of variability by person and maybe by age.
Yeah, exactly. We had started with the Amish longevity study, so that we have samples from centenarians, their spouses, and their children. And there is an incredible amount of variability, like 3 or 4-fold in terms of nucleotide excision repair capacity that we're detecting. We do have a long way to go to understand if that has a functional impact, but I think we're all surprised at the extent of variability we're discovering at this point. I guess I would raise a question for the participants in this. We all come from very strong DNA repair backgrounds. So is anybody thinking of something that we have not mentioned that we should be trying to link between DNA repair and a certain disease phenotype or other phenotypes? If anybody wants to drop something in the chat or speak up, we're always looking for new applications, and we come from a narrow perspective.
Good point. Good point.
Yeah, I think that's that's kind of the feedback that we all are looking for. Of course, a lot of you guys are working with Mary Beth as an epidemiologist, and a lot of the projects have clinicians and all that. But what we're looking for is kind of wider feedback on what would it take, if you were considering some of these assays, what would you want to see to be able to fit these in to your ongoing studies, or maybe even a new study? Is it kind of ease of use? I think a lot of times things become possible. For example, DNA methylation became possible on a large scale with Illumina sequencing and methodologies that got the cost and the throughput to a point where it was really doable on a large number of people. What I assume, it's the driving factors are going to be kind of cost and throughput, but are there any other kind of feedback from you all, and we're early on in these development of these methods, but what would it take for you to really be able to use it? And I think in Mary Beth's and David's case, they've really shown they can do this in very small amounts of blood that they irradiate, and can look at the ability of the cells to kind of deal with that double strand break damage. That's fairly high throughput. But for the rest of you, I mean, are there any comments on, as you think about measuring, repair capacity in your studies, what would you really want to see before you would be comfortable applying these methods?
And Dan, I'm sorry,
Michelle,
Rob, go ahead.
And then I'm going to have
Gwen also posted an important point about cost, and I haven't, but maybe some of us should start thinking about how much would it cost per sample, for example, moving forward. That's a good point.
And then maybe I'll just build on what you just said, Rob, just thinking about Laura's invitation for collaboration, and maybe that collaboration doesn't need to be a full-blown, study, but maybe it is just a little pilot project where you're kind of testing, you have an inkling of, hey, I think this might be useful, and could we do just a smaller study? So maybe just kind of opening it up to both the DNA repair experts as well as the epidemiologists here that are, thank you for your time to be able to help inform us on this.
Right, and Gwen's comment in the chat was basically asking if any of the developers have tried to monetize the assay yet, to show the epidemiologists the range of costs to consider when planning collaborations. I don't know if we have a sense of that yet. I mean, since we're all kind of, a lot of you are at the very early stage of development. Any sense, projection of, for example, for the UDS assay or for FMHCR, like cost per sample, or is it too early to tell?
Yeah, I could say for FMHCR, it's a moving target, because one of the things we've been doing is figuring out how to make the reporter plasmids more quickly and more efficiently, and we've made a lot of progress on that. So that was the bottleneck. And I think at this time, it's in the range of $100 or a couple $100 per sample to get this, at least a 4-plex assay done with our most difficult to produce reporters. So it's going to fall in that range.
And Rob, I think with the multiplex, they're expanding to already to 384 well plates for COMET assay, that's become incredibly significant savings on doing samples by COMET.
Yeah, it seems to be working, at least in our initial. We're trying to finish that study up now, and the challenge is, we sort of had to make a makeshift mold, right, which we did manually, but probably cost about $100,000 to actually make the real mold, of which we can't afford in the grant, right? So that would have to come afterwards, but it seems to be working. So it would certainly would reduce it, because each chip costs about $250, right? So if you could limit the number of chips per 10 samples or something, that would bring the cost down considerably.
And Mary Beth, I would assume, since you've got all of the irradiation equipment and capillary tubes and everything worked out, it isn't terribly expensive to run a lot of samples through David Brenner's assay.
That's right. And he's actually been doing that for, during emergency response. He's gone over to Japan to do things, even just with blood spots. But yeah, I don't want to speak for him. So, but I'm happy to share his contact information if anyone wants to, or if you email me, I can connect you through email to Dr. Brenner. Yeah.
Great. So let's see. Jacob, do you want to just unmute and ask your question, or I can read it either way?
Yeah. So I guess across all of the talks, one thing that I would like to know before considering using any assay is how reproducible it is across duplicate samples. So, just because it produces a value, I would want to be certain that it's consistent when we use it multiple times or measure it on the same sample multiple times, we get similar values.
Yeah, this is something I think we're all thinking about to varying degrees. And one of the things that that's come up for me is that there are a lot of ways to think about reproducibility. There's, as you mentioned, within a sample, one of the challenges with the primary samples is you can't necessarily hold that sample and then measure it again. We can do technical replicates, but we see also that there may be some day-to-day batch effects. So we've looked at taking multiple blood draws from the same person or freezing down several tubes from one blood draw and then making repeated measurements on those. But we also think that it's possible that within person, there's temporal variation, could be driven by lifestyle, circadian effects, the environmental exposures, whether the person is sick or not, kind of coming back to this reverse causality question. So I think some of us are still trying to decide what's the gold standard for establishing the reproducibility. What type of measurement would be most compelling to you, having asked that question? Would you want to see a within-person stability or the same sample coming out the same way over and over again? Or is there another way to assess that? How have other people accomplished that with assays that are more, widely used?
So I think within person is particularly appealing to me. So from your talk, I really liked how you looked at the temporal stability of the samples over time. And that's evidence to me that you showed temporal stability. So you might suspect that if you took the same sample measured at the same time point, that it would also be stable. So that was one thing that I found very encouraging from your talk. But some of the other assays, that would be something that I would need to be convinced about before, moving ahead.
I would just comment on that too, that we're working really hard on this stability over time, reproducibility within individuals, freezing, thawing, all sorts of variables that might affect it. But right now, my biggest problem is quality of samples. So if we're trying to get fresh blood, that we've just seen an incredible amount of variation in sampling. And any ideas how to standardize that would be very helpful too.
Well, I mean, I think there's a couple different things. I think that's the ultimate challenge, right? Epidemiologists, it's easier to do field sample collection from populations and then freeze them and have them come in. This fresh blood that needs to be processed pretty quickly is, while it is ideal for a lot of these flow cytometry and other components of the work that's happening, it also is a huge barrier to being able to sort of access the, to implement this in sort of large scale population-based research, unless you have everybody come to the clinic and you have your phlebotomist on hand along with your lab folks on hand to be able to process right away. So those are some of the things I think of as potential barriers for kind of scaling up some of this work, and because those are limitations in that regard, I think, right? And so anytime as an epidemiologist you can have a standard protocol that's really well monitored and measured over time, that's one way to improve standardization. That's pretty well. One other question that I have around some of these assays around reproducibility and stability is also response to environmental exposures. And I'm wondering how much work, and I apologize, I was super busy, but I find this topic really interesting. So I haven't listened to all of your talks. I'll go back and do that. But just to be truly honest about where I'm at with all of this. But I think one of the things that I'm wondering is how much research in sort of non-human samples has been done to test sort of viability, exposure response time. So if you're looking at what does that look like from an environmental perspective, and how much of that basic work has been done. So, translation going back and forth between animal human models, in vitro assays, and then the human samples themselves for some of this.
Well, for the COMET assay, I mean, this has been done at the very small scale, at the single cell, the single COMET assay has been going on now 30, 40 years. So there's certainly a large cohort. And then the in vivo COMET assay is even a standardized test around the world. of course, not the Comet chip though at this point. But so from that perspective, there's a lot of information that I think is understood in terms of what it means to get that signal. I think the biggest challenge has been mentioned is the samples that we get. Like, so we, in this particular study, we have our own phlebotomist, and the people have to come to a certain location, which of course now with COVID makes it even more challenging. We get pretty much 50% cancel rate on a weekly basis, but it still gives us that opportunity to isolate everything. So we get in our case, we get the blood tubes within 2 hours of isolation, and then we actually process them ourselves. And that keeps that standardization a little bit more careful. I agree with Jacob and other questions about reproducibility and stability, and these are the things that we're evaluating.
And Laura, I think you mentioned that you've done a number of studies with mice with different NER defects and things like that. So I mean, it's fairly well characterized in terms of even the data that you showed in your talk about knocking out a specific NER protein, and I think a lot of that has gone into the development of the assay initially. What do you expect to see? I think one of the interesting things that's come out in the fundamental research is the effect of circadian rhythm on NER. But it kind of goes back to Kristen's point, is that the ability to kind of collect things at the same time of day from a large patient population or population-based study is probably pretty low. I mean, we probably just have to factor a lot of these variables into how you interpret the results. Any thoughts, Laura?
Yeah, the circadian rhythm really unnerves me a little bit, because we do have to pay attention to that. And we're hearing this even from, pulmonologists who are taking bronchoalveolar lavage samples at different times of day. There's an incredible literature on that. So I think we're going to have to incorporate that at the next iteration of thinking about our assays and our SOPs for sample collection. I think it's critically important. I haven't even dared to do that in mice yet. But their circadian rhythm is backwards, so maybe that's not necessary.
And Dan, I think that's an important research question for thinking about how to give cancer drugs, if that's the case.
Yep. Very important.
Yeah, I think this is one of those things that on the one hand, it's fascinating. And then on the other hand, as we're trying to make optimal assays, it's terrifying, because we're trying to hold as much constant as we can. And then once we've established the assays, then I think we'll be perfectly positioned to study some of these questions. One of the things on our project is that we're doing measurements at the time of lung cancer diagnosis, and then if possible during therapy, and then later if people go into remission. And that, I think, will give us a sense for, that's a pretty large environmental impact, because they're undergoing radiation therapy. So it's a defined exposure, as well as a change in health status, to the extent that the cancer is going to be present at the time of diagnosis and not in remission. So we're really eager to see how big the differences are there. I think that'll give us a sense of kind of the magnitude of within-person variation that's possible with major events that might impact what we can measure.
And Laura posted something kind of estimating the UDS assay at about $20 per sample, and then up to $50 if you have to analyze those kind of separately. So, I think we'll get there. A lot of times, these assays, when they get to the kit form, get a lot cheaper to do, and that's probably a ways off. But again, it's been very helpful to hear some of your initial comments, on these assays as they're being developed. That it's a very important kind of input. Anyway, this has been great. It's maybe a strange way to do a workshop when you don't have the presentations fresh in your mind. But you go back and look at these in more detail and you have questions, reach out. You could send your questions to me directly at NIEHS. My email is Shaugh1, SHAUGHN1 at niehs.nih.gov, and I'll direct them to the developers. But as we go, we're always interested in your feedback, and we think these are really exciting kind of new approaches that really I think will help not only in kind of population-based studies, but as Mary Beth and others have said, these could really be used in cancer treatment, in looking at individual or precision medicine kind of efforts. So again, thanks to everybody to the presenters for making the videos and being here for questions, and then for all the participants and your comments.
Thanks, everyone.
Thank you.
Thank you to all presenters and all attendees. Thank you.
Yeah.
Thank you.